Okay, so we left off talking about the role of the pancreas, and why are there subtitles? Okay, so when we left off, we were talking about the role of the pancreas and starting to look at the role of the small intestines and how that is where the majority of digestion is occurring. And so now we're going to look a little bit in more detail at the structure of the small intestine. And so if we were to zoom in on an actual intestine, if we take like a cross section, we'll notice that we have these different layers. And especially on the inside, it looks like you have these multiple finger-like projections with structures that are called villi. And so what you'll be seeing a lot of are transverse sections of that small intestine, and your job will be to label the diagram and know the different layers. So this whole top layer, which includes the villi, is called the mucosa. And the villi are these finger-like projections, and we'll look into more detail at the structures of those. And just below this mucosa, you have a submucosa, and underneath that, you'll have your muscle layers. Remember that the small intestine has both circular and longitudinal muscle, and that the circular muscle is laying on top of it in this really wide layer, and just underneath that is the longitudinal smooth muscle. So if we look at different cross sections again, the villi and what is just below it comprises the entire mucosa, and the villi just refers to those hair-like finger projections, and then the submucosa is underlying that original mucosa. And again, in the muscular layer, the first larger one is going to be the circular muscle, whereas the longitudinal muscles will be along the bottom here. So again, just another view. Villi, hair-like projections, whole thing is the mucosa. This underneath it, this first layer is the submucosa, and then these are your muscle layers. The larger one on top, circular muscles, the one underneath is the longitudinal. And it's a little bit clearer here. Again, that whole dark region is your mucosa, and then this region here where you have those hair-like projections, that's your villi. Underneath that first mucosa, you have your submucosa. And in this photograph, the circular muscle layer is very clear on top of that longitudinal muscle layer. Okay, so just another labeling there for you. Okay, so if we zoom in to the villi, so these are the teeny tiny villi here. We'll see that it has a even more complex structure, that what you're really looking at is a layer of epithelial cells, and we'll be looking at in detail what's happening there, and that we have two main structures in the villi, the lacteal, which is where fatty acids are going to be absorbed and transported, and then you have these blood capillaries, which are going to be where glucose and your amino acids are absorbed and transported around the body. So again, just another look at that, we have epithelial cells along the edge, lacteal in the center, and those blood capillaries on either side. So as digestion is taking place, remember that your polymers are being digested into those monomers. So that's by enzymes like amylase, lipase, and proteinase. And those polymers cannot cross these barriers. They have to be broken down into the monomer size in order to be able to cross. So let's take a step back from there to review some of our membrane structure. So remember, the main components of the membrane are the phospholipids, and remember they are amphipathic in nature, so you have those polar hydrophilic heads facing the outside and inner surface of the cell. You've got things like cholesterol that are helping to maintain fluidity in the membrane itself. And then you've got these proteins that are traversing the membrane, things like your protein channels. And so this is an integral membrane protein because it integrates into the membrane. You also have your peripheral proteins that are going to be off to either side. And then you've got things like your glycoproteins. So these are just proteins that have some sugars on top of them. And this was what comprised the entire membrane structure. So again, just a reminder that phospholipids are amphipathic. The heads are polar because that phosphate has a charged oxygen group on it, and we have those hydrophobic fatty acid tails. So again, just a reminder that cholesterol, and again, this is what that lipid looks like. It's got a really complicated ring structure, and this is sitting in the membrane really close to those polar heads. So this is found only in animals. It does control that membrane fluidity, and it can help what diffuses through. And so cholesterol is going to come into play when we talk about our lipids, right? So let's review, while we're here, some of our membrane protein functions. So some of those integral membrane proteins are hormone binding sites. So insulin, when it's going through your body, it's finding insulin receptors and binding to those receptors to allow that cell to uptake glucose. The enzymes that we've been talking about in the small intestine, they're actually immobilized on the lumen of the intestine. So this has their active site exposed. They can be used again and again from where they are. The channel proteins, right? So this was what was partly involved in passive transport. So only specific types of molecules can move through these channel proteins. And then we have pump proteins, which was active transport, where we're creating a concentration gradient in our cells. Okay, so the transport through the villi is going to involve multiple types of transport, including simple diffusion, facilitated diffusion, exocytosis, protein pumps, and co-transport. So we're going to quickly review our different kinds of transports. And so just a reminder that our two main classes are going to be the transport of fatty acids through the lacteal and the transport of glucose and amino acids through these capillaries. And so let's take a closer look at the actual structure of this villi. And so everything from this point on, we're going to be focusing on these little epithelial cells. And so this is your main cell, and you've got the microvilli on this side. So this entire structure is the villi, is the villus. And so we are focusing for our purposes on what is happening inside the epithelial cells. And so these epithelial cells have a top and a bottom, right? And so they have characteristics that you're familiar with. They've got a nucleus, they've got rough endoplasmic reticulum and Golgi and mitochondria, and their whole job is to facilitate the transport of the monomers that are being created inside the small intestine, inside the lumen of the small intestine, through that apical surface and out through that basolateral surface. So digestion on this side is happening inside the intestine. And so this is where those enzymes that were created in the pancreas, like amylase and lipase, they're actually immobilized on top of the apical part of the cell, where they are then allowed to digest those polymers into monomers. And then, so let's look at glucose transport specifically. So this involves both active transport and facilitated diffusion. So glucose will always have some kind of protein involved. And just a reminder that with active transport, you're going from a low to high concentration. It does require ATP, and it will create a concentration gradient that we usually use for energy to do like a facilitated diffusion. So just a reminder that facilitated diffusion is passive. And in this case, instead of moving from low to high, you're moving from a high to low concentration and no ATP is required, that these are specific for certain molecules. So yes, we have two types of cellular transport. Passive does not require ATP, whereas active does. And just to remind you, we have three different versions. Simple diffusion, which does not require any kind of membrane protein. Facilitated diffusion, which does require a membrane protein. And then you also have osmosis, which is following the movement of water. Okay, so onto glucose. So the transport of glucose, again, involves both passive, facilitated diffusion, so I'm using some sort of transport protein, and then also active transport in the form of a protein pump. And so this is the summary of how that happens over here on the right, but we're going to go through this step by step. So here I have my epithelial cell, and I have the apical side, which you can think of the top side, which faces the lumen of the intestine, and I have the basolateral side, which faces the blood vessels. So the first part of this is getting glucose into the cell to begin with. And that happens through a sodium glucose symporter. It can also be called a transporter. And how this is working is it's using the energy of a gradient of sodium to move glucose. So, and this is a symporter, a co-transporter, meaning that sodium and glucose only travel together. You might know some of this from like your Gatorade, right? Gatorade has both salt and sugar, and that is in part because of this glucose symporter, that sugar and salt like to move together. So this is passive transport, and specifically it's facilitated diffusion because it is using this channel protein to move that glucose. And that transport protein is called SLT1, so like solute transporter 1. And just a reminder that passive transport only moves things from an area of high concentration. So in this case, the high concentration of both the sodium and the glucose would have to be high on the outside, and it would have to be comparatively low in the inside in order for this to work. And so for that to happen, we actually first have to create a low concentration gradient specifically of the sodium. And so to do that, remember, we've got these active transports, right? So we have things like a protein pump, which will move from low to high. So will exocytosis, where we're exiting proteins that we generate inside the cell to outside, or endocytosis, where we're bringing things from the inside to or from the outside to in. So what we're going to focus on is this protein pump, specifically the sodium-potassium protein pump, because back here, I have to create a low concentration of sodium. And the way that I do that is through the use of a sodium-potassium transport, which we have looked at before. And so this sodium-potassium protein pump is now working on the basolateral side because I want to create low sodium inside the cell. And so what this is doing, right, so I'm having potassiums transported in. So these two potassiums be transported in, and potassiums and three sodiums are being transported out. So when these three sodiums are being transported out, that's giving me a low concentration of sodium inside. And so that is what is creating that low sodium concentration that's going to allow that co-transporter to move both the sodium and the glucose. It cannot work if there is still a high concentration of sodium. The last part is now getting the glucose out. And so part 3 is, again, we're back to facilitated diffusion, where we have this glucose uniporter. And so again, this is passive transport. As the glucose is coming in, we're creating a high concentration gradient of glucose in the cell. And so then it's no big thing for that glucose, to go back out and follow that facilitated diffusion pattern. So what this basolateral cell is facing is facing the blood vessels. And so these blood vessels are going to go all over the body, and there are a couple destinations that glucose can go to after that, but we'll talk about that in a minute. So this is basically the entire overview of glucose transport. So remember that inside the intestinal lumen is where you have amylase breaking down that starch into maltose, and then you have maltase breaking down that maltose into glucose molecules. And so step 1 is that glucose will find a sodium and go through that sodium glucose cotransporter for facilitated diffusion. So this is passive. But this one will only work if there's a low concentration of sodium inside that epithelial cell, which is created by the sodium-potassium protein pump, which is kicking sodiums outside the cell to generate that low sodium gradient to allow this cotransporter to work. And that the third part then is taking the glucose that's come in and then getting it out through that GLUT2 transport protein, which is facilitated diffusion, and then that is going into the blood where it can be distributed to its last destinations, right? So there are 2 places that glucose is going to go after it is absorbed through those epithelial cells, and one of them, right, we know from our study of the pancreas that the pancreas are secreting insulin, and that is going through the circulatory system and telling cells, hey, you've got glucose out here, go ahead and take it into the cell. So once you have that glucose in the blood, it's going around to those different tissues, same as insulin, and hanging out in that bloodstream until insulin is coming along and telling those glucose molecules that they can be transported into the cell. So that's one option, right? So if you don't have, if you have low blood sugar and your body knows hey, I need to get energy in these cells, it's going to go directly into the blood and go supply that energy to whatever cells that need it. If, however, you have an excess of blood sugar, you don't need sugar to burn right now, you can store it in the liver. So instead of going to the regular circulatory system where you're going all over the body, if you have excess glucose, it's going to go through the hepatic portal vein and get stored in the liver as glycogen. Right, so this was one of our polysaccharides, and so this is the one with the alpha 1,4 and alpha 1,6 connections. So it takes that glucose through the hepatic portal vein and links them together in that glycogen where they can be stored. And you, I think, store up to about 24 hours of glycogen in your liver that you can use at a time. Okay, so the capillaries transport glucose and amino acids. Now we're going to focus on the lacteal and how that transports fatty acids. So just a reminder, again, okay, we have our membrane here. Reminder that the membrane, the inside, is hydrophobic, and that we have this cholesterol molecule in here which is also hydrophobic. So, and again, reminder that it regulates what diffuses through. So let's look at our triglycerides. So just a reminder that the fatty acids are hydrophobic, right? So they have those long fatty acid chains, and because the membrane is hydrophobic, a fatty acid will have no problem crossing this membrane. And what it does is it crosses the membrane and it actually combines with cholesterol. So from there, right, we combine with cholesterol when we get in. From there, it actually reassembles into a triglyceride, and then it goes into the Golgi apparatus where it combines with proteins to form lipoproteins, and then via exocytosis, right, because that's what the Golgi does, it exits proteins into vesicles, and because those lipids were attached to proteins, they get to come along on the ride with the vesicle, and so they are being secreted via exocytosis, and they are being pitched to the lacteal, right? And so they enter the lacteal, your fatty acids enter the lacteal, and they are carried away from the intestines. So just to kind of review a little bit, that we have in our intestines, right, we have these structures, and the main loopy structure is called the villi. And if we look at the villi, we've got the lacteal, which is where the fatty acids that are absorbed are going to go. And then we have the capillaries where the glucose is being transported, and all of this is being facilitated initially by these epithelial cells that have an apical face and a basolateral face, and that once, say, the glucose is inside, it can either go through the blood vessels to the hepatic portal vein to the liver to be stored as glycogen, if that's what's going to happen to it if you have an excess of glucose, or will go throughout the body through the circulatory system to supply glucose to those cells. So that's it for the digestion for 6.1.